This is a video demonstration of the WNERR larval fish eDNA protocol for larval toes. First thing that you're going to do is get a big plastic bin, fill it up with 10% bleach solution. You usually estimate. Then I'm going to put the larval toe net into that solution, submerge it completely. Make sure you get the cod end in there and the tow rope, anything else you want to be sterilized. Wait for 15 minutes, come back, and you're going to start to rinse everything off. You're going to want to be super adamant about where you place your sterile net. That's why I bleached the wall where I'm going to put it. Pour out that bleach solution. Don't ruin your clothes like I have a million times, and hang up that bleach net. Really what we're doing right now is we're trying to set up the net so that it's totally sterile, because when we get to the harbor, we want to make sure that everything we collect and everything in the water sample that we take afterwards is really from that toe and not a previous toe. I'm going to rinse off everything. If there's anything still on there that's not been killed by the bleach, it's going to be collected in this water sample that I'm about to grab. You can see here the eDNA kit that's been prepared beforehand with a Nestle Pure Life water bottle. I'm going to fill up water bottle just by dipping it in. Make sure I have clean gloves. And once you have it capped, just tip it over and pretend to squeeze it a little bit. Make sure nothing gets out and everything's secure. All right, we're ready to take our harbor samples. So I'm going to be the sampler today, and Michelle is going to be the assistant. That's a great way to prevent contamination in the field. I'm going to pour out the water in that water bottle. We're having fun there. I'm going to submerge it using a grabber tool that you see in my hand, putting it down 1 meter to get water from there. And that's the same depth that we put our tow net later on. So it's pretty cool. We take that water back to the lab at UNH and extract the DNA, and fingers crossed, what we find is the same species that Michelle can taxonomically ID in the lab. The true goal of this project is to be able to go to the harbor, take a quick grab of water, sequence that in the lab, and say, hey, here's a snapshot of the biodiversity in our harbor. And here's the real fun part. We're going to spray off this net. All the little pieces of vegetation, all those little plankton are going to get washed in down to the bottom of that net, and the really small ones, as well as just tiny, tiny cells, are going to get washed down into that really dirty rinse water. So the sample we're taking is of the rinse water that's been touching everything, all these things that got caught in the net over an entire hour. Another thing that we can do is unscrew that cod end that's collected all of the biomass and pour it into a sterilized blender. We can blend that up until it's a homogeneous solution, take a subsample and sequence that in the lab as well. The Wells Reserve has been conducting traditional larval tows at the same location for 10 years. Back at the lab, research assistants pick through the samples to find and identify larval fish, crabs, and invertebrates. We can compare the identified species from traditional larval toes and eDNA toes that were taken simultaneously, and these similarities and differences can tell us a lot about the efficiency and accuracy of our methods.